Welcome to Octorum, the literature review for the obscure scientist. My name is Isabelle Berthelot, your host, with Oliver Hutchins, my co-host. Stay tuned for more. Hi, everyone. This is Isabelle welcoming you back. It's been quite a while. Apologize about that. But there are many reasons, and we would love to share that with you. Oliver, do you want to say hi to the people?
Hello, people. It's been a hot second, but I'm glad we're all here again in this auditory space together.
Yes. So there has been a pretty big gap between our last episode. And actually, what y'all don't know is we did sit down to record many episodes. We did do a full length podcast about flowers, and that was a really fun conceptual episode. We ended up not publishing it because it was a little bit different than what we normally do. It was not based in academic literature. We didn't include any publications in there. It was purely a conversation, and it was awesome. But we figured maybe it's a little too different, a little too soon. And so we ended up not publishing it. I think the last thing that we published was in October. I think it was that Halloween episode. So it has been a long time. Both Oliver and I have had many exciting life changes, and we want to share that with you. So, Oliver, do you want to start with some of your exciting news?
Oh, well, I would love to. So I can't even remember how much my personal life I've shared with the podcast. But, I used to work at a botanical garden where I did some plant research in various labs and whatnot. I lost that job due to funding circumstances, was just unemployed for a few months, saying, will I ever work again? Then lo and behold, a beautiful opportunity to do some plant restoration work in South Africa has emerged, and I'm working on putting that together right now. It isn't all completely locked in yet, but if this goes as we're planning, this could be year one of a multi-year project in building the capacity to do ecosystem restoration in the savanna of South Africa.
Yes, so exciting. And for those who may have forgotten, you previously did work in South Africa. So you're returning to a collaborator that you were previously with, right?
Mm-hmm. Yeah, so most of my research experience happened in the plant ecology lab that happened at this research station in Kruger National Park. It was about 4 years between then and now, but then we came back in contact and we're like, we got to do more plant stuff because we're like the only two guys who have cared about anything other than trees and grasses in that park for about the past century. So there's a whole bunch of stuff that only we want to do. And now other people are kind of interested too. So we're going to be learning how to grow the wildflowers, the grasses, the trees, just all the plants of the savanna at a research capacity, just so we can build mass restoration. So I'm feeling pretty good about it. It's going to be research. It's going to be practical. It's going to be translating research to decision making, which is kind of like my favorite place to be in. Like I don't want to do something deeply fulfilling that then just sits in a university library archive for decades and only 8 PhDs even read it, and 4 of them say they don't like it. Like 4 of them are just reading it so that they can tear me apart. I don't like those scenarios, so I'm happy that this is going to be something where it's going to be translated to real changes in the world. Fingers crossed.
Yeah, absolutely. And definitely with your work in environmental science specifically, there's a lot of people who have a lot of anxiety about the future in terms of environmental science. So I know that you have a lot of thoughts and feelings about the restoration work, and I think it's great that you're going to be able to be hands-on, actually making a difference. Feel like I remember you saying that there was going to be a pretty heavy connection between your experience seed banking at the botanical garden that you were doing, and propagating rare plants, and the work that you're doing in South Africa. Is that correct?
Yeah, so if we want to get into the nitty gritty of it a little bit, which I always do, I just try to spare the ear of most people who get stuck in a conversation with me. It is going to be learning how to grow things from seed. So the way I explain it to people is if you're saying, oh, I want to start a little veggie garden, and you're starting stuff from seed, you can go to a garden center, buy a little pack of tomato seeds that says, grow these at this temperature, in this level of sunlight, do all this stuff, and it'll grow 90% of the time. All of that information came from decades of scientific research and then centuries of agricultural built experience and wisdom. manifested in that little info blurb on the back of that packet telling you what to do, and just the production and availability of those seeds anyway. And that's for, a tomato, like one of the staples of modern gardening agriculture. If you're working with species that nobody has ever tried to grow before, which is pretty often with, wild species, a lot of people just say, yep, that's nature, it's there and part of it, and then never say, oh, what if we tried to grow it? So a lot of these times, if you're in the restoration space where you're saying, hey, we want to take this land that used to be pristine ecosystem and try and make it back what it was again. Plants are the foundation of pretty much every ecosystem. You have to start there and then hope everything else will come back. And you can't bring the plants back if you don't know how to grow them. So there's that very specific niche of seed expertise that all restoration can't really happen without. And that's the nice little niche that I perfectly fill after working in a conservation seed bank for about 2 years.
Yeah. So I'm really excited for you.
Mm-hmm. But Isabel, like what I'm doing pales compared to what you are about to do. Like, why don't you tell our lovely viewers what's going on in your life?
Yeah, I mean, I wouldn't go that far.
It
I
pales.
think the way that you talk about planting wildflowers for the elephants in the savannah of South Africa, I mean, that's beautiful. So I wouldn't go that far, but I am really excited. As many of you know, have been working as a medical research specialist for over a year. Recently wrapped up my master's degree in pathology. I talked about it a little bit on that unpublished episode about flowers, but this is sort of a side tangent. I've also recently started working in an eye bank. It's connected to organ donation, and so that's been a really interesting experience for me as well. I am really interested in terms of research, organoid technology, and so it's been really cool to see um, potential ways that that could be connected to transplant research and to clinical work. But that's been a really interesting experience in and of itself. So that's been another reason why I've been personally really busy over the past few months. But I also was recently accepted into Tulane's biomedical PhD program. So I'm really excited. I'm actually going to be starting in June. So that should be really fun. And with that, with the time that has passed, with my future planning of becoming an independent scientist myself, I have been doing a lot of reflecting on the type of scientist that I want to become, the types of strengths that go with that lifestyle and with that skill set. But I've been thinking a lot about curiosity, how to foster it, the role of this podcast in doing that. I've also been talking to other people who are in the field of science. I had a wonderful interview with one of my friends who's doing his PhD in physics and nuclear physics at MIT, and we discussed sort of the role of a scientist. And Oliver, I would love your thoughts on this because I feel like it has contributed to sort of a reset that we're doing with this podcast right now. We talked about it a little bit in that archived episode, but sort of finding your identity in science. Science can be really daunting because it's about forming new ideas, new conclusions, new experiments, and sometimes it feels like if you're not bringing something really significant to the table, then what's the point? And I feel like that does lead to a lot of burnout, and so that's an interesting thing to consider. One thing that I wanted with this podcast, and I know that you've talked about too, having this podcast to be a space about simply intellectual play, making connections, asking questions. And one of the things that we wanted to get across with this reset for this podcast is this is a fun thing that we do to strengthen our minds. It's not like we're sitting here trying to have the answers for things for you. It's really not that at all. We're just exploring work that previous scientists have done, providing questions rather than answers. Because that's sort of the main muscle that needs to be built if you're going into science. But Oliver, I just wanted to know if you had any thoughts, any reflections on your role as an individual, as Oliver Hutchins, in the world of science, if you bring something unique to it. It's been things that I've been thinking about a lot, and I certainly have thoughts on it for our listeners who might be in the same boat and wondering.
Yeah, I completely understand what you're saying. So I have a long answer to this that I think will eventually answer your question. So the way I think about it, the role you can play as an individual under kind of two frameworks of role and identity. The first one is just realm of expertise in terms of, you know, are you a biologist? Are you a chemist? Blah, 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 blah. And even once you're within that discipline, what's your tiny little niche that you narrow down more and more and more, or not even narrowed down, just like what realm do you establish? Are you someone who just takes a single grain of the world and say, this is my everything? Or do you remain more of a generalist and say, this whole realm is where I must be? And I think, there's that whole spectrum of existence a scientist can be on. The other dimension of that is, all right, even once you've decided, this frame of knowledge that you're building your career or life around, what are you doing with that knowledge or that data? Again, whatever you want to name it. Are you generating it or are you disseminating it? And I think that exists on, a sliding scale spectrum where on one side you have nothing but creation, no dissemination. You're a chemist for a private company whose just job is to just publish papers. You don't care where it goes or what it does. You're just there because you love it and they're paying you good money for it. On the other end of that sliding spectrum, I see is just science communication, where you might not be doing any research, you might not be finding anything new about the world, but you're saying this knowledge needs to reach more people. My role is to make as many people realize this as possible without pursuing new information. So I am always thinking of, realm of information and dissemination of information as a little framework of how you want to navigate that. The other thing I think of here, the role of science as a whole, is I return maybe about once a week to Simone Weil's definition of the role of science, in which she said, in Gravity and Grace, a beloved book that we both mutually obsessed over, she says all knowledge has really just three applications: industrial application or technical application, a game of chess being that kind of exercise of the brain that you're talking about, and a road to God, she says, are the three things that knowledge can offer. So I'm kind of going back and forth between how I perceive the role of science and how Simone Weil perceives the role of science, and asking myself, how do I want to respond to that? For myself personally, I don't care as much about generating new knowledge. I kind of see that as inevitably that's just a part of the career, but that's not what it's really about or the focus to me. I really see the role of dissemination of what we already know and then refinement of that. Because I think of, or that's just how I think of restoration, in both that pursuit of God or spirituality, again, however you want to name that, and the dissemination of that. Because, especially thinking of restoration, the most popular opinion about the world right now seems to be people think we destroy everything that we touch. We only make everything worse. The only thing we can do to improve that is take this hands-off approach and say, we're not going to have anything to do with the natural world. Which is just doomed to fail, of course, because we're a species on the earth just like anything else. There's no exclusion scenario, and any attempt to do so just hurts everyone involved. And two, I feel like it ignores the really phenomenal possibility, knowledge, and research that already exists, saying these systems that we used to think once they're gone forever is not true at all. There are huge swaths of the world, of ecosystems that if we just make a genuine attempt and refine our process, we can do unimaginable work to make the world a better place. One of the most common stories I feel like I hear in conservation restoration is, one, the deep grief of seeing what's happening to the earth. But one of my favorite ones is hearing the people who say, I thought I would spend my career watching this thing being lost, but over the course of it, we've learned how to save it. And whenever I think like, what do I want to do? I don't want to refine a little equation mapping population dynamics for the rest of my life. I want more people to see that, because I feel like that would transform people's perception of the state of the world. And a lot of people would say, well, that's not science. Or if you think about kind of like the broad societal perception of what science is, you're thinking of a guy in a lab coat, like mixing chemicals between beakers. That feels very different from that. But I think it goes back to that same heart of the, I'll say, curation of knowledge and just the importance of that for the broader world.
Yeah. And I do have a follow-up question. With different pathways of what you can do in the field, where do you look to decide where you fit in or what you want to be like? Do you search internally or are you looking at examples of people that you look up to?
Mm. It's a little bit of both. I feel like I'm never intentionally I'm going to see it this way or I'm going to see it that way. I'm going to look for others. I'm going to look within myself. I think the process, without me being too self aware of it, is probably a mix of both. I feel like the exciting thing about science is that if you're really on the cutting edge looking forward, you don't find that many people who are experts or know where you're going. Like usually if you find the people who are having the same conversation, even the experts are not standing that much ahead of you. So I feel like even if you ask them for advice, they just say, I have no clue, but do you want to do this work with me? I don't think there needs to be as much planning or intentionality once you have that narrowing down of expertise, that realm of information, of once you have the sense of where you want to be. The second you identify that and start talking to that people, you get that amazing sense of just so much to be done with the information that we already have. You just have to fill that vacuum space. Because, I mean, whenever I think about the role that I'm playing right now or where my career is going, this isn't a result of me saying, seeds in the savannah, this is my everything, this is who I am, this is the only way to make it work. It never looked anything like that. I've always just been kind of dancing around, playing different roles that were all vaguely botanical. I've done some plant ecology work, I've done plant conservation, I've done horticultural landscaping. They're all kind of within that conversation. But there was never anything that said to me, this is the only way. I don't know if that happens to anyone. I think there are some people who genuinely do have that recognition of, this is my specialty. I think there are a lot of people who, in that quest for identity, convince themselves that this is their thing. I can't really say or speak for anyone else. For myself, I haven't found that sense of this is what it has to be. But just by virtue of getting this handful of experiences, knowledge sets of working in all these different realms, playing all these different roles, it gives me a combination of skills that all kind of push me towards one direction of playing a very specialized role without even very much intending to. It just kind of like was the natural flow of things. I think you just kind of have to start walking in that direction and just narrowing down as you go.
No, I very much understand what you're saying, and I think that I value the same thing in terms of curiosity about everything. I think
Hmm.
that to me, the way that I feel about science and the people that I talk to who I feel like are the most passionate about science, they all share this idea that everything is all connected. So if you're interested in science at all, it's usually everything. It's never like a particular thing because everything is just a web and it's all interconnected. With this future step of beginning my PhD, I have been doing a lot of reflecting on what it means to be a great scientist, not necessarily a specific thing that I want to research, but who I want to be.
Mm.
Um, and so I've been looking towards reflections of people who I believe are great scientists and great thinkers that I look up to and ways that I can strengthen and train my mind to think like a great thinker, but also in a way that is uniquely me. So I did a lot of reflecting and soul searching for this sort of identity in the world of science. I think that one thing that I was struggling with was losing myself in the pressure of needing to achieve something and forgetting to find myself truly in the curiosity. And so with these next few months before I start my program, I'm really trying to focus on that. And once again, with that, I want to emphasize that this podcast for me, and I know for you, it's something fun. Like I am not even a I don't even have my PhD yet. I don't want people to listen to this and think that I know everything. I'm in an early period of training. And so I don't want people to think that I feel like I already have it all figured out, but I do want people to think that I'm trying to figure it out. And that's where I'm at in life right now. I don't have things figured out, but I very much am on that journey and am excited about that journey. And so this, what you're listening to right now, it's just an attempt. It's practice. That's all that this is. And I think that there's a lot of value in that, but I also don't want people to mistake it for me being an expert. I'm not an expert on anything, but I do love to practice because I do love what I do. But anyway, like I said, I was struggling with the pressure, but then I decided to sort of release that and find peace and curiosity. One thing that I've done for myself is I started a ginormous garden. I'm growing like one of everything, and it's just given me so much purpose and going back to that like childlike wonder about how things work because after all, that's how I got started in science. It was simply looking at the diversity and watching the ways that all of these different plants and insects and everything just behave together. And so I've been trying to find myself in that. Instead of asking a question every time, I started shifting my focus towards expanding the opportunities to seek data rather than having to have having to know what I was looking for whenever I was going into a situation. And reflecting on the people that I believe are great scientists, I feel like it's always broadening your world, opening doors, and being in the present, and also having a mindful understanding of new technology. Like, to me, that's what creates a good scientist. It's actually finding new ways to acquire more data, but not necessarily having a specific place where you know that the data is going to fit in. Um, so opportunities to seek data of any kind. I believe that this can happen in the lab, but I think that this can also just happen in life. It's something that I've adapted to my life. If you want different data than other people, find different places to look. Like what I was saying about starting my garden, I wanted to experience something different. And so I just looked in a different place. And I feel like the same can be said, there's a lot of pressure in life about having exciting experiences and stuff like that. And truth is, you don't need to like go skydiving or something to be interesting or to have an interesting experience. You can just do things that not everyone else is doing. And I feel like, especially in the world right now, I'm sorry if this is like a long run on sentence rant, but I do believe that these things all connect. I feel like in the world right now, we talk a lot about automation and we talk a lot about trends in the masses. And I do think that a lot of people are having very similar experiences in life. And I feel like some of the stress of life and of science and of patterns of thinking might be coming from the fact that a lot of people are doing the same thing. And so I want to challenge and train my brain to just go to a different setting, find a different piece of technology, find a different way to get different data. It doesn't have to be a specific question, just different. And I would urge you guys all to do the same. And Oliver, I would love to hear your thoughts on this too. It's a very inventive state of mind, entrepreneurial state of mind, and thinking about the role that I want to play in the world of science. I feel like that is sort of where I fit in. I feel like my personality, I value creativity and I value entrepreneurship. And so I feel like this has helped me become more comfortable with myself as I'm starting this journey, which I'll tell y'all a secret. I'm scared. Like, this is something new and like I'm nervous. And so I feel like this has just been a step for me.
I think the best way I can link myself to everything you just said is what's becoming one of my central life philosophies is just process over product. And I think that's at the heart of it for everything to me, of just all your love and passion must be directed towards just the process of actually doing it with no thought about what comes out of it. and whatever comes out of it is either a transactional receipt symbol saying this happened, or it's just icing on the cake. So, whenever I think about this, I think of all of life's truths in the form of gardening and plant care and stuff like that. A lot of people go in saying, if I'm going to grow an orchid, it's like, I want to see this thing bloom. Like, that is what I'm doing this for. That's the goal. How do I make that happen? Assuming this is a difficult process for you, which it will be because you're trying to learn how to do something new with something that hasn't been made to be easy to do, you're just going to get frustrated and exhausted and mad that you aren't getting what you want to get. That's what happens if you direct your attention and love towards the product, i.e., this orchid in bloom. But if you take all of your love and direct it towards the process, saying, man, I love watering plants. Man, I love mixing up special soils and going on weird websites where I have to order special orchid soil amendments just to make their roots happy, which I did this week. It was the highlight of my week. You would need to love saying, man, I love making sure, I love seeing sunlight, like the right kind of light, hitting those leaves. Man, I love picking weeds. That's the big one. Like, so many people just say, I hate weeding. And I feel like if you can love all of that, you never even have to think about the product because that just inevitably comes because you've been investing everything properly in the process. So I think that translates to how I'm starting to see science too. I think it really needs to be redirected back towards that process of looking differently, looking more deeply, doing the work itself, not even thinking about what it's going to look like in the future. And I think to require or to do that, you're talking about how it's a scary process for you or, just jumping into something new. I think inevitably to not look at the product is a leap of faith in just the process. You want to have what it's going to be before it's even there, just to, immediately reap the fruits of your labor before you've even done the labor. And I think that's just a quest for certainty, giving yourself what you already think you want instead of opening up and saying, why don't I just do something different and then see what happens? So I think that's my understanding of play, curiosity in the realm of science as I've come to understand it for myself.
And I feel like right now I'm trying to remind myself, you were talking about falling in love with the process.
Mm-hmm.
I'm trying to remind myself that looking at things from a different perspective is what I love. And so always finding like a different angle to look at something, and that can include a piece of technology that gives you a different view. And that's why I'm really excited for this episode, just to transition to the content, because I feel like that's what this episode does completely. And I'm super excited to get into it because, of course, everything that we talked about, we can sit here and talk all day, but it's not meant for just us two. We try to do these episodes for our listeners. So we have to ask ourselves, how does what we just said relate to our listeners? How can we take this paradigm shift of not asking specific questions, but considering new places to gather data from, new technologies, new angles, and we can practice focusing on the creative methods and the deep, long thinking behind them. And that's what we want to do. Hopefully, as you're listening to this episode, and maybe in the coming weeks and months, you as a listener can begin asking yourself, what could be a potential technological tool that gets us different data from a different lens? After all, science is, at least in theory, very coherent. There might be millions of different units putting out millions of different readings, but the idea is that they all contribute to one coherent story of life, like the same scene being viewed from the naked eye or from a microscopic lens or from a polarized lens, night vision goggles, whatever. The idea is that it's the same thing just being seen in different ways. And sort of what we're saying is like, what if you came up with a completely new tool to view the same thing? For those of you who know me, you know that I love art. I'm an artist. And I love it for similar reasons that I love science. It's because it's an amazing way to see how one thing can be portrayed in so many different ways. the way that a great artist stretches the mind of how one object, thought, or emotion can be perceived, comprehended, and expressed is the same curiosity that drives researchers to seek out information. I want to play a little mind game. Remember in art class, whenever the art teacher would put an object in front of you and say, draw this, and you would begin drawing it, only to realize at the end that what ends up on your paper has much more to do with what your brain says the object is and not what it truly looks like. And then the next day, the art teacher puts down the same object in an attempt to dissolve the assumptions that your brain has about the object, says, draw this again, but using only positive and negative space. No lines, no gray, just black and white, positive and negative space. And after this exercise, you look at the paper, almost an abstract lava lamp form of black and white that, wait, actually ends up being more accurate than the original depiction that you started with yesterday. This is where we are. We're trying to make sense of life and what we see. So let's pretend that we draw life, the universe, with only positive and negative space, only black and white.
As you were saying that, I was imagining my kind of like freshman year art class where you just put like a cube on the table and was like, draw this cube. I don't know if my cube can compete against the
Well,
universe in
what if
black
it
and
was
white.
something living?
Oh, it's like,
And the teacher was like, capture its aliveness, only using positive and negative space.
oh, okay.
What I would expect someone to say, or like after they have had a long time to consider it and like draw it or whatever, I would assume that this seeming void of space and air would be black, dark, empty, negative space, with the objects all around us, the people that we loved, carved in solid white positive space. And why is that? Because that's sort of what this artistic exercise is meant to capture in terms of the negative and positive space. Why do we do that? Well, because density, firstly. If something is more dense, there is literally more matter per volume, and more stuff is more positive space. Objects and people are all matter. But what's funny about this concept, the visual of the white tangible matter with the black void background, is without active containment of this order, this structured matter, the universe would just seek equilibrium, and your artwork of black and white would just be a solid sheet of the perfect mix of gray.
Mm.
And so I sort of use this mind exercise to make us aware that life is so amazing in the sense of it retains its order even when the universe is seeking out energetic equilibrium. The reason why we can visualize the world as it is now with positive and negative space is because of containment. And that's sort of where this episode is going. It's called Order, Chaos, and the Barrier in Between, where we're discussing this wonderful quality of life by truly looking at how order retains its form in the world of chaos. In that black and white, positive and negative space picture, lines, distinctions between positive and negative space must emerge. There is no black and white without the line that separates the two. And in this episode, we want to honor the line, the border, the barrier that separates order and chaos.
As I was reading like all these papers, I started thinking of an exchange in the book The World Religions by Houston Smith, which is this great little book that's broad overviews of different religious perspectives from around the world. And in there's this exchange where the author's talking to a monk. I can't remember what type of monk he was. I think he might have been like Thai Buddhist. The author was asking a monk what he thought God looked like. And the monk said, like, I've been trying, but I don't think I can, quite get my head around God. But the closest I've been able to come to him was imagining an endless sea of gray tapioca. And that image has just kind of like struck with me, or has sat with me ever since I read that book. Like, since in like freshman year of high school, because I think my first thought there was just, that's the most boring God I've ever heard of. It's just like I don't want that to be God. I'm not a fan of the humanization or anthropomorphizing of God as like dude in a robe with a beard. But I think I still prefer that over endless sea of gray tapioca. But I think that gets to that same thing you're talking about of what happens when you remove all the boundaries and borders and just everything becomes kind of that the whole universe would just be grayness. I also think about that in, how if you mix all the colors on the color spectrum together, you get that kind of like muddy, gray, brown kind of color, where it's just, you think that everything at once would be the supreme version of diversity and everything, but it's kind of like the loss of everything at the same time. And I think the question there then is, if we don't have barriers, does that mean we have everything, or does that mean we have nothing?
That's a question that I'll have to think on for a long time. But
Mm,
what I certainly know is you would not have anything living.
mm,
So yeah, that's an important question to think about. But also, whenever living things break down, they release everything.
mm,
This episode is so exciting to me because my particular focus and research does happen to be barrier function, but truly, barriers are such the underdogs of life. Without enclosure, without separation via a barrier, there would be no such thing as life. Life is a bubble of order that is actively maintained in a chaotic world. And I want to share some thoughts on the value of barriers and the relationship between thermodynamics and biology, as described in the wonderful paper Biology and Thermodynamics Seemingly Opposite Phenomenon in a Search of a Unified Paradigm by Shahar Dolev and Avshalom Elitzer, published in the Einstein Quarterly Journal of Biology and Medicine in 1998. Here's a short excerpt that I will read for y'all. Open quote. Notice first that the most illuminating demonstration of thermodynamics' pertinence to the life sciences comes from observing the processes to which organism is subject upon dying. All of the manifestations of decay that reduce living tissue back to inorganic ashes share the fundamental physical characteristic, namely complying with the second law. The decomposing organism goes back to the state of equilibrium, thermal, chemical, etc., with its environment. Being alive, then, means being far from equilibrium with the environment, thereby manifesting the autonomy which is the very hallmark of life. Later, they write, even when no movement is apparent, the living body fights entropy all the time by performing enormous microscopic work. Ion pumps keep the right concentration of ions across the cell membrane. Various enzymes check cell structure and the DNA strands for errors. Membrane proteins convey nutrients in and waste out. Complex systems cooperate to keep homeostasis, etc. It is these intracellular processes that later converge with amazing precision into macroscopic movements. No less striking from the thermodynamic viewpoint is the coarse development of a single creature, its ontogeny. An oak tree, for example, begins its life as a zygote smaller than a millimeter. Within a few years, it consumes basic chemical elements from the surrounding air and soil, elements from highly disordered sources, only to organize them into the form of a mature, ordered, highly complex tree. Life has the unique ability to act against the normal course of events. End quote. I particularly like this review article because it captures the awe of how active life is, actively going against gradients and taking the chaos of the environment and turning it into usable energy, then using that energy to continue shielding from the chaos. And this effort results in an organism that grows, lives, but ultimately returns to this dispersed, disordered natural state. This review does a great job in giving this visual of order and disorder in a broader picture of life, that positive and negative space, the black and white illustration. But let's reflect on how the role of life is to maintain an ordered state, distinct from the entropy of the universe, and this inherently means separating itself from the universe through some form of containment, some shield, membrane, or barrier. We see what life and death means in this black and white illustration. So we see that when this maintenance is faulty, when the separation and barrier gives out, there is death and disease. For the mental play in this episode, I want to tie life, death, and disease to simply changes in the barrier, inspecting the strength of the floodgates that are constantly holding back the chaos of the environment. Oliver, do you have any thoughts on this?
Yeah, I got some stuff to say. You bet it, buddy. You should never have allowed me here. So, I mean, just my comment on the paper as a whole. I think it lands. That title is just bait for us. The seemingly opposite phenomena in search of a unified paradigm. That was basically written for us. Let's take these things that seem contradictory and unite them into something bigger. I saw that and I was like, yes, who doesn't go crazy for a unified framework? Yeah, but thinking more specifically of how you're connecting that to the notion of a barrier is just, as I was reading this paper, I was just thinking very broadly, even outside this specific context of biology, of just what a barrier is, just kind of sitting with the word. And I think my immediate, more emotional reaction to the concept of a barrier is this sense of denial, rejection. Nothing is happening, nothing is passing through. And the more I thought about that, I was just trying to think when is that true in which there's a case in which there's just truly no movement? I started asking, is there ever a case in which we come across a barrier that truly lets nothing through? Because, even the tallest walls in the world allow birds to just fly straight over, allow air exchange. Might not be able to go over, but everything else can just fine. And so that led me to start perceiving barriers as this kind of paradoxical mechanism of movement, where we often identify barriers fairly quickly by what they prevent from happening. But once we start looking closer and more holistically, we can see barriers are really about what's allowing to happen. So in this sense, it's just, plugging beautifully into what you were already saying of just, you'd think the setting of barriers and boundaries would be antithetical to the existence of life within the universe. But what we're starting to see is that there cannot be life without these setting of boundaries.
Exactly. Whenever you do look into what barriers actually look like in science, they are very complex. They're often selectively permeable, not impermeable, and that is essential to their function and it's essential to their role in life. Whenever I think of barriers that are essential for humans, the first thing that comes to mind, skin, for example, tissue, and then, of course, which this is unified in all of life, but cell membranes, organelle membranes, these are all examples of essential barriers that regulate life. Like actual floodgates, they're made up of machinery as well. With this perspective that all disease and all death can be boiled down to disordered barriers, then this machinery becomes essential in understanding life, death, and disease. So, where do we go from here as scientists? Our job is to, of course, consider concepts and questions to illuminate new knowledge. And I feel like this is one thing that we've done really well with this podcast in our previous episodes. But I also feel like this podcast hasn't succeeded in building up the other essential ingredients of a good scientist, which is gathering data, ideally in novel ways. And so for this episode, I want to explain how scientists in the real world, working in labs and hospitals, anywhere, outdoors, we have one paper about plant science, actually measure barrier function as well as comprehend it. Part of the beauty of science is connecting these larger, more conceptual questions into tangible forms of technology that we can use to measure and collect data. One of the pioneer thinkers for barrier function analysis was Don Powell, who wrote Barrier Function of Epithelia in 1981 for the American Journal of Physiology. And just as a reminder for our listeners, the epithelia is the outermost layer of tissues and organs. Your skin is covered in epithelial tissue, but also your internal linings like your digestive tract, your respiratory tract, your glands like your thyroid, your organs, etc. Usually, this epithelial layer is the outermost layer because it has created a barrier for the structure, but it is also specialized to absorb nutrients and even further specialized to secrete, whether that's mucus, sweat, enzymes, etc. So, sort of what Oliver was saying, it is dynamic. It takes in and it puts out and it divides. Because we know that barriers are critical for the functioning of an organism and each respective organ and tissue, the ability for the epithelium to properly contain the inside and separate the outside is a key factor of life. This containment is conferred by junctions in between the epithelial cells. There are many different molecules that create this junction, connection, seal, but probably the most commonly discussed is tight junction. To put this in perspective for you, tight junction barrier loss is a key topic in the digestive tract and is a key player in why inflammatory bowel disease, Crohn's disease, ulcerative colitis is so damaging. And this also happens to be the focus of a lot of my research. Barriers are especially important in the digestive tract because this is one of the few areas in the body in which the other side of the barrier will actually contain bacteria and pathogens. In these inflammatory conditions that I just listed, when a barrier is lost, bacteria can also pass through the epithelial wall and cause infection or inflammation, which is the body trying to fight off disease. That can be even more damaging than the infection sometimes. If you know anyone with celiac disease, this is a disease in which barrier degrading molecules are released after consumption of wheat and grain, and the opening of this barrier wall in the digestive tract is what causes such severe reactions. There are also specialized immune cells that will attack this protein found in gluten and actually damage the epithelium of the small intestine, causing issues in nutrient absorption as well. So you already know about barrier function, you just might not realize it. The last example that I'll give is one that is not specific to the digestive tract because, like I said, epithelial tissue and therefore tissue barriers exist all throughout the body. Sepsis, which is the leading cause of hospitalizations and death, 20% of global deaths from the CDC, that's a lot, is ultimately fatal because of barrier loss. Many people think that sepsis is due to rampant infection, but that's actually not the case. It's the body's extreme reaction to infection that causes the issue, but it's also why it's so neglected, because people don't often know that it's happening when it's happening. But ultimately, the body has a catastrophic immune response that also damages tissues in an attempt to kill the pathogen. The key source of damage here is when epithelial barriers in the vascular tissue and organs become damaged. What happens when your vascular tissue no longer contains blood inside? Plasma starts leaking out of the vasculature, causing edema, which is fluid buildup in the body, and you have massive and life-threatening drop in blood pressure. So back to us as scientists. If we want to study these diseases and study barrier function, we need to have a way to measure it. Dan Powell's barrier function of epithelia identifies a quantifiable characteristic of barriers. I was going to say something stupid like, can anyone guess what happens? But I feel like that's stupid.
Like it's Dora the Explorer. It's like, do you see the membrane? Me too. Just giving people like 4 seconds.
Yeah.
That'd be really fun. That'd be fun.
If you can guess what aspects of the barrier can be quantified, well, epithelial barriers need to absorb nutrients on the outer side, which we call the apical lumen, not really important, and also secrete nutrients into the inner side of the body on the basolateral membrane, as well as selectively allow for movement of nutrients across the barrier between the cells. The nutrients in question are molecules. Ions are molecules that have charge. Part of the structure of barriers is not just to protect matter from entering or exiting, but by allowing the careful movement of ions and separating these electrical charges across the membrane. Doing this can harness energy. This harnessed energy by separating electrical currents across the membrane is also how cells in the digestive tract are able to bring in specific molecules. The absorption of glucose in the digestive tract is essential for living. That's why we have to eat. And glucose, being such a large molecule, requires energy to get into the cell and energy to pass through the cell and inside of the body where it can enter the bloodstream. If there were to be a break in the barrier, not only would matter, like bacteria, potentially pass through, but we would also lose this electrochemical gradient that allows us to extract glucose from the lumen. So, when we're trying to assess barrier function, how well a tissue can separate and distinguish inside versus outside, we use something called transepithelial electrical resistance, which measures the electrical signal from the outside of the membrane, then the inside of the membrane, and then calculates the difference. I want to share this research method with you because it's a great way to understand the value of a barrier. If there is no transepithelial electrical resistance across the barrier, this means that the electrical signal is the same inside and outside of the barrier, and there is no distinction between the two. This follows the same fundamental thermodynamic understanding that we talked about. If the disorder within a system is the same outside as inside, this is biological death. It's loss of active preservation of order that is so unique to life. It really makes me wonder what our research into disease and pathology would look like if we focused heavily on the resistance and integrity of the structures that enclose and separate, whether that's skin separating our whole body from the outer world, epithelia separating one region of our bodies from another, cells separating intercellular space from extracellular space, or even organelles separating from the cytoplasm. It is all a gradient, and it is all a tool to preserve order.
I have something that I don't really know where it goes, but something I want to play with. I get. We need to have like a little like ding. It's like this is a play moment. It's like all the kids listening and be like, "Yay!" Yeah. So one thing I've been thinking about this whole framework of understanding sickness as the breakdown of barriers. I was kind of going into the opposite direction sometimes as I've just been thinking about it of can we also understand sickness as the tightening of barriers sometimes, or just like the closing off saying we used to let something in, now we're letting nothing in because we're in crisis mode.
Yeah, like looking at it from a perspective of restriction rather than like dissolution of the barrier.
Mm-hmm.
I would still classify that as dysfunction. And sort of, I think, I mean, I'm not sure about this. I would need to double check, but similar to what I was saying about the electrochemical gradient, that has so much to do with the function of barriers. Because barriers, like I said, are more than just this mechanical wall. It's actually functional. It's dynamic, like what you were saying.
Mm-hmm.
And so whenever there is something that prohibits the movement of ions, the movement of things that are selected to go across the membrane, then that causes dysfunction via restriction. And so then you don't have nutrient uptake. Or if you, like, to go back to what you were saying conceptually, closing oneself off is still dysfunction. It's just of a different kind.
Oh, okay, yeah. So you just go into just any high school AP biology talking about the cell as, just this perfect basic unit of life. They talk about, what is the little bubble that allows this whole thing to work? It's a semi-permeable membrane, which I remember in, that first high school class, I was like, what's the point of having this boundary that protects this entire living system that's semi-permeable? You want to hear that it's this like rigid skeleton as hard as diamond that like allows life to resist despite all odds. But no, it's built into that understanding of, if if you cut the cell off from the rest of the world, it would just suffocate and die and turn into nothing. Like, there needs to be some amount of exchange with the rest of the world so that the cell can get what it needs and get rid of what it doesn't. It's just cool having that perspective shift of going from seeing the strongest boundary as a non-permeable boundary to realize the most foundational boundary is kind of like the softest, most rubbery. Some things can literally just pass straight through.
I think people take it for granted, like what you were saying in your class, and you learn about the cell membrane, blah blah blah. People just think that it's just, the normal anatomy of a cell. People don't realize if you lose that membrane, if there's like one hole poked in that membrane by bacteria or something, you're done. Like
Mm.
it's over. It's so essential. A cell becomes dead whenever the membrane is penetrated and it can't contain the energy anymore. It's dead. So I
Mm.
think that people take that for granted. And this paper by Dan Powell reviews significant real-world connection between changes in barrier function during exposure to alcohol, biosalts, antibiotics, etc. This connection paved the way for future studies that connected exposure of tissues directly to their loss of function, measuring it with transepithelial electrical resistance. The last paper that I want to discuss is the role of barriers on a cellular level, not just the tissue level. We know that every cell in an organism shares the same genetic code, and diversity and specialization of cells comes about by selectively silencing and enhancing the very long shared genetic code. Epithelial cells are specialized cells that look different from other cells. One reason why they look different is because they have one side of the cell that faces outward of the structure and one side that faces inward. This asymmetry is called polarity or polarization. Without polarization, there would be no organization within the cell to, for example, bring glucose in one side and pass it out the other, effectively absorbing nutrients. Polarization isn't just a magical shape that the cell takes on. It's regulated by barriers within the cell that separate one end of the cell from another. Cell polarization is such an important aspect to the maintenance of life, and we don't realize that it is regulated, meaning if some damage were to occur or if this polarized cell became dysregulated, this polarized specialization would be lost along with the cell's ability to absorb nutrients. Cell polarization is relevant in other aspects as well. And when cancer cells take over, they also hijack cells to polarize them so that they can squeeze through inaccessible areas to metastasize and spread. For this reason, it's important to understand how to measure polarization of cells. In this way, cell polarization shows functional barriers within cells, an example of how barriers can be functional rather than just physical. Dysregulation of these functional barriers contributes to disease and ultimately can result in organismal death if the dysregulation is severe enough. Once again, because I want to explore research methods in this episode, I want to share a paper by Yan Gong and colleagues, published in the New Phytologist, titled Quantitative and Dynamic Cell Polarity Tracking in Plants. This paper outlines the development and application of polarity measurement, an image analysis technique designed to quantify cell polarity, in this case in plant cells. This paper automates cell polarity measurement by adding fluorescent dye to image the cell membrane and the plant-specific polarization proteins, then calculating the relationship between the two via fluorescence intensity and confocal imaging, which is basically a microscope that can perceive different colored lasers from the dyed molecules. This quantitative method allows for faster and more accurate polarization measurements that surpass subjective assessment. Although this demonstration is specific to plant cells, this technique can be adapted to animal cells as well, and scientists demonstrated this by using the PAR protein, which is the polarization protein in animals, in C. elegans.
I really love cell polarity because I think this is where that concept of a functional barrier really shines. Whenever, you first think of barriers, you think about like the physicality and the strength of them. But, this is a step up from just saying, semi-permeable. This is just thinking of barriers, that void space, the absence more than anything. And that works better than I thought it would. Because, whenever I think about, like the idea of like, a 30-foot stone wall, the functionality of a wall as a barrier isn't due to the presence of, the stone as much as it is about the absence of people where that wall is. So even in like the most concrete barriers I can think of, that barrier is still about functional absence of whatever it's regulating. So it's cool to see the polarized cell as an example of that. I forgot, did you mention just kind of like proteins within a cell's framework of polarization?
Yeah, I mean, there's proteins all around the cell, and in this case, the actual functional part of a polarized cell has to do with the channel proteins on either side, but there are polarization proteins. In this paper, they image specifically plant-specific polarization proteins, but then humans have different ones, the PAR proteins, that basically act as like a compass, if you will, that determine where either side of the cell is going to be designated.
Mm-hmm. I think that's so cool to just say, like, if you're looking at how cells orient them in space and in reference to the rest of the body, to think that there's just these little compasses made of nitrogen that allow the cell to get its bearings within the world. Like, as I was reading this, I was thinking of just the North Star, just in astronomical navigation, this tiny little blip of light in the sky that's just stable and steadfast enough that we can build our whole world around it. That's how I've started thinking about these polar proteins, of just these little blips of nitrogen that allow the cell to know, all right, this one's over here, this one's over there. We can triangulate and build the rest of our body around that framework. I think that's super cool. And again, like if those, two polar regions are what allow this cell to make sense of itself, that's a really powerful boundary, and it has every incentive not to violate that or conflate that boundary.
Yeah, and polarization itself, like I said, it's not a magical thing. And once again, it is something that we take for granted because in certain diseases and whatnot, that polarization can be lost. And whenever the polarization's lost, that means that, take, for example, the epithelial cells of the digestive tract. That means that it's not going to be able to absorb glucose and release it into the bloodstream. You're not going to be able to absorb nutrients whenever you eat. That's going to cause disease. And so people don't realize that just because something is an epithelial cell doesn't mean that it's going to be perfectly polarized all the time. Whenever disease occurs, that can change. Ultimately, form confers function, and when we look at the form of life on all scales, microscopic cells or whole organisms, we see enclosure and containment time and time again. This barrier is the wall that separates chaos from order, and the structure itself is what life is. In a world of black and white, we often forget that there is a thin division that holds these two polar opposites apart.
If you're talking about what should people take away from this conversation, this episode, how should people be thinking about barriers going forward, I think a nice little logical framework is whatever barrier you're up against, either that you're intellectually just exploring or whatever barrier you're coming across in your own life, it might be cool to ask these two questions. Question one just being, what is the function of this barrier? And number two being, what is allowed to cross and what isn't? All right, everyone, thank you so much for joining us for episode 7 of Octorum. Reminder that we have an active Instagram account called Octorum_podcast. You can also find us on our website, octorum.com, and subscribe to our newsletter there too. We never crowd out your inbox, we just send out emails when we publish a new episode. If anyone is interested in hearing our archived episodes about flowers, no scientific literature, just poems and thoughts and conversation, please let us know and we will publish it if there is demand. Thank you to all of our followers and fans. Talk to you next time.